Guys, it's Miss Faye, and today we're going to be covering DNA replication. By the end of today's lesson, you should be able to define replication, describe the process of replication, identify when it occurs, where it occurs, and finally, why does it happen, the purpose of DNA replication. Before we get started, I want you to keep two things in mind. First of all, DNA is the information that must be replicated in order to maintain continuity. In other words, in order for cells to be able to reproduce and produce and make more cells, in order for organisms to reproduce, their DNA has to be copied in order to ensure that the daughter cells that are produced contain identical copies of the DNA. Remember, DNA is the blueprint. It essentially carries all the information that tells us everything we need to know about the organism, that makes the organism who they are. This information is then going to be used in order to build proteins. And proteins, we've already learned throughout this entire year just how important they are as molecules. They are going to play a role in virtually everything within the organism's processes, in the cells, etc. They basically determine all of the traits that we see, all of the phenotypes that are present and we see visibly in all of the organisms are determined by the presence of proteins. Now, if we were to kind of look at the sequence of these terms, how are they related to one another? DNA carries the information, and this information is kind of written as genes. So think of genes as the individual letters that come together to build a recipe. Okay, so DNA is like the whole recipe book, and the letters that are present in this recipe book are the genes. Now, the genes are going to be used, so these instructions are going to be used to build a protein which will determine a trait. And remember, every trait is determined by one protein. So DNA contains genes, which are the instructions for making proteins, and finally, proteins determine the physical phenotypes or traits that we see. Now, what is DNA replication? If you remember from our talk about the cell cycle, we know that DNA replication is, if we had to simplify it, it's just copying DNA, making copies of DNA, taking the DNA, duplicating it, replicating it, making a copy of DNA. And this happens during S phase of the cell cycle. So if you remember, the entire cell cycle has different stages or phases. DNA replication is happening right here in the S phase. And it takes place inside the nucleus of eukaryotic cells. And the reason why we say eukaryotic here is because if you remember, prokaryotic cells do not have a nucleus, right? So replication will not happen in the nucleus of prokaryotic cells. In prokaryotic cells, replication happens in the cytoplasm. However, for our cells, for eukaryotic cells, we have a nucleus, and that is where replication will take place. Why does replication happen? What's the point of making copies of the DNA? This will assure that every cell has identical DNA, identical genetic information. So every daughter cell that is produced is going to have identical genetic information to the cell that made it, okay? And this also allows the daughter cells that are produced to have identical copies of the DNA that was present in the parental cells. Again, this maintains continuity. So in order for this whole process to happen, we're going to need a template strand, okay, and a complementary strand. What do we mean by a template? What is a template? So a template is a model that you use to create other things. So for example, sometimes we're working on a project in class, and I'll give you a template. Like let's say you have to do a infographic or a brochure. Instead of me telling you here, create a brochure from scratch, I can give you a template of that brochure, and then you can use my template in order to customize it and create your own. All right, so part of the work is already done for you. Same thing with DNA replication. So in DNA replication, we will have two strands. A DNA strand, okay, and that's the original DNA strand that exists within the cell, and a complementary strand that's going to be created during the process of DNA replication. Now, during DNA replication, the rules of base pairing are going to apply. And if you remember, what are the rules of base pairing? What does that mean? Let's go back to the Chargaff's rules that we talked about. Adenine is always going to pair with thymine. Cytosine is always going to pair with guanine, and so on. Okay, so these are what we mean here by base pairing rules. So that's going to apply during DNA replication. So we start off with the DNA strand, the original DNA strand, and remember, it's a double helix, so it's going to have two sides. But the first thing you want to do is you want to split that double helix open, separate it, okay, and then once the strand separates, you're going to build a complementary strand. So in this example, the original DNA strand is shown in blue, and the complementary strand that has been created during the process of replication is shown in red. A complementary strand, just like the name suggests, complements the original DNA strand, and it complements it by following the base pairing rules. Let's take a look. The original strand had the following sequence of base pairs: adenine, guanine, adenine, cytosine, and so on. The complementary strand basically complemented, right, because of the base pairing rules. So adenine paired with, as we know, thymine, guanine paired with cytosine, adenine paired with thymine, cytosine paired with guanine, and so on. We're following Chargaff's rules in order to build a complementary strand for the original DNA strand. Now remember how I said that the original, because it's a double helix, the original DNA strand has to separate? It's going to look something like this. Okay, so this is kind of showing you the exact same thing, the exact same process from a different angle. Again, this is the original DNA strand that came from the parental cell, and you'll notice that the base pairs are held together in the center. Now, what bond is holding these bases together? Let's kind of rewind to when we talked about DNA structure. What is the bond that is present in the center of a double helix? Hydrogen bond, okay? And then what are the bonds found on the edges? Covalent. And if you remember, I said keep that in mind. We're going to refer back to it. Here we go. The reason why it makes sense to have hydrogen bonds in the center is because hydrogen bonds are a lot easier to break. Remember, they're weaker bonds. So since you have to unwind and then eventually open up, unzip, just like a zipper, unzip this double helix, it makes sense to have hydrogen bonds in the center because they're a lot easier to break than covalent bonds, which are found on the edges, because you don't want to separate the bases from the backbone. Okay, so you break these hydrogen bonds, you unzip, you separate the parental original DNA strand, and then you start to build complements, or you start to build complementary nucleotides in order to build the complementary strand, just like we saw right here. Okay, notice that at the end, okay, I want you to keep in mind that the original parental DNA strand and the two strands that are created at the end of DNA replication are identical, which is exactly what we want. That's the whole point of replication, to make identical copies of the DNA so that this here goes to the first daughter cell and then this here goes to the second daughter cell. All right, so remember that nothing goes on in our cell without the help of something, okay? And in this case, we have two very important enzymes. Obviously, there's more enzymes, but for our purposes, we want to focus on two very important enzymes that are going to help with the process of replication. The first one, protein or enzyme, okay, remember all enzymes are proteins, so that's why I'm using these words interchangeably. The first enzyme or protein that is helping the process of replication is helicase. And helicase is what's going to unzip, break these hydrogen bonds, open up separate the original strand. Okay, that's going to be helicase. The second really important enzyme we want to focus on is DNA polymerase, and that is the enzyme that's going to add the complementary bases during this stage right here, or during this stage right here. Okay, it adds the complementary bases, and it also has another very important role. It proofreads and makes sure, double checks there's no errors that are happening when you're using the base pairing rules. Okay, that's about it for today. I hope that was clear, and as usual, if you have any questions, please let me know.